for sure.
Okay, I think we can get started. So thanks everyone for coming to the Marine Science Seminar this Friday. I'm happy to introduce our speaker, Patricia Thibodeau. She is currently a postdoctoral fellow at University of Rhode Island, working on an NSF EPSCoR consortium project. She received her PhD from the Virginia Institute of Marine Sciences in 2019, and also has a bachelor's from Bowdoin College in Maine. And she was also a Hollings Fellow with NOAA Fisheries in Seattle. So she is already well published in her research field. And I was also really impressed by some of her efforts in using art in science communication. And I recommend checking out her video called Plankton Patterns on YouTube. But today, I think she's going to be taking us to Antarctica and back to Rhode Island. So with that, let's welcome Patricia.
Great. Thank you so much, Dr. Hayes, for that introduction, for inviting me. We actually found each other on Twitter, which I'm like so much a proponent of Twitter now for networking and everything. It's great. So I'll go ahead and share my screen. See, can someone give me a verbal yes they can see?
We're getting the notes view.
Okay.
You swap it.
Yep.
This happened to me in class yesterday. There we go.
Yeah. How does this work?
That looks good.
Great. Okay. My gaze will kind of be shifting between two screens here, but yeah, so thank you everyone for being here today. I'm excited to talk with you all about plankton time series and all the great research and data we can get from them. I'll be talking about time series from the Southern Ocean, down in Antarctica, all the way up to Narragansett Bay, back where I am here in Rhode Island. And I'm currently presenting to you all from the University of Rhode Island. And I wanted to acknowledge that I'm here on Narragansett lands. And today, as I mentioned, I'll be talking with you about time series, which reflects research I've done as part of my PhD, as well as my postdoc here at URI. And throughout my scientific career, I've had the opportunity to analyze extensive time series data sets and ecological time series. And these time series are particularly valuable when we're focusing on plankton because of their short generation times and limited mobility, which can make them great indicators of long-term change. One aspect of change that time series help us to understand is temperature. And while most of us know that the Earth's been warming, I really like this image here that shows the temperature anomalies since the 1900s. And so we have the global mean temperature up here on the right, and then we have the temperature anomaly here for each country in the world, and we have any bar below the zero line here indicating below average temperature, and any bar above the zero line indicating above average temperature. And we can see that right around the 1990s, the bars get longer and redder, and particularly when this ends in 2016, we see that this temperature increase has been really pronounced. And just to orient you here, we have the United States in this red arrow, and we can see that it has one of the biggest and reddest bars in the world. And one ecological response to this long-term warming are shifts in phenology. And phenology is the study of annually occurring life history events. And the New York Times has a really great illustration of phenology. This image here shows the timing of spring foliage in 2017, and we can see that it begins in January down here in the south where you all are, and then begins in May all the way up in the Northeast and Pacific Northwest. And when we compare that spring foliage timing to the 30-year average spring foliage, we see that in the south, the spring foliage is occurring up to 10 days later than it used to 30 years ago. Whereas in the California coast and Pacific Northwest, we see that spring foliage is occurring up to 10 days earlier than it used to 30 years ago. And this brings me to the time series work I've been working with, which enables me to both think about phenology and long-term change in regions of warming. And so I'll be talking with you about the Narragansett Bay, which is a temperate estuary that's been experiencing long-term warming. This figure here shows mean winter temperature on the x-axis and annual temperature on, annual temperature on the y-axis, year on the x-axis. And just put my laser on. We see that there has been a long-term increase in temperature of 1.4 to 1.6 degrees centigrade over the last 50 years. In a very different region of the world, in the Southern Ocean, we've also been seeing long-term warming along the Western Antarctic Peninsula, which is across the Drake Passage from Chile. And this figure here shows year and winter degree temperature on the y-axis. And we've seen a long-term increase in winter temperature, an average of 1.1 degrees centigrade increase per decade, which has represented a 7 degrees centigrade increase over time. And that represents five times that of the global average. This region has also been seeing declines in sea ice. This figure here shows sea ice duration in days over time, and it's been declining since the 1980s, such that we now see an overall 3-month decrease in sea ice season since 1979. And what's fortuitous is that in both these regions that are experiencing long-term change, there have been time series that enable us to document how these changes are affecting the ecosystem. So in Narragansett Bay, we have a long-term plankton time series. And then along the peninsula, we have the Palmer Antarctica Long-Term Ecological Research Program. And I've had the opportunity to work with both of these time series, and it's enabled me to ask the following questions. First, what are the effects of climate change on pteropod ecology and physiology along the Western Antarctic Peninsula? And pteropods are these beautiful open ocean snails that I will talk about more in a moment. And second, what are the long-term changes of phytoplankton abundance and bloom phenology in Narragansett Bay? So focusing on this first question, I'll be using pteropods as a model organism, and pteropod in Greek means winged foot. And they're commonly called sea butterflies. And this video might give you a sense as to why. I hope it's coming through clear on your end. People tell me they often look like little sea bats, or I think creatively look like the snitch in Harry Potter. But they are also a very ecologically relevant and important group in the Southern Ocean. This species that I'm showing you here is called Limacina halicina antarctica, and it's the third most abundant zooplankton along the peninsula after two kinds of krill, the Antarctic krill and another species. And one of the reasons that you may have heard of pteropods before is because their shell is composed of calcium carbonate in a more soluble form called aragonite that more easily dissolves when the ocean waters are more acidic. And so they're considered bioindicators of ocean acidification. But within the Southern Ocean, they're also an ecologically relevant group because they're important grazers of phytoplankton. They have some of the highest grazing rates among zooplankton in the Southern Ocean. This is a figure of a different kind of pteropod called Coralla, but it's one of the few images we have of a pteropod feeding in the open ocean. And most pteropods use a mucus web, which you can see the strands of here. And then basically any particle that is small enough to be caught in this web is collected and then retracted into the pteropod for it to feed on. Pteropods are also an important prey for higher trophic animals, especially fish and seabirds along the peninsula. But when I was beginning my PhD, I realized that we didn't really have a very good understanding still of their general ecology and physiology, especially in the Southern Ocean. And so this led to my overall research goal of determining the environmental controls on pteropod ecology and physiology along the Western Antarctic Peninsula. And this led to three primary research questions. The first being, are there long-term changes in pteropod abundance and distribution related to environmental controls? And this work was published in LNO in 2019. And then, have Limacina phenology patterns shifted over time, and what environmental conditions control these patterns? And this was published in Deep Sea Research last year. And then how do changes in environmental conditions along the peninsula affect Limacina metabolism? And this work was published in GIMBY last year as well. And so focusing on this first question, are there long-term changes in pteropod abundance and distribution or biogeography related to environmental controls? I utilize an extensive data set that's been collected as part of the Palmer LTER. And every year since 1993, a group of scientists has embarked on this big bright orange ship called the Lawrence M. Gould. And we traverse from Chile down to the tip of the Antarctic Peninsula. And then we spend about 30 days doing 24/7 science, collecting a suite of biological data at each of these grid stations shown in white. And what we're really interested in trying to understand is this north to south climate gradient that's developed and how that's affecting the ecosystem. Because the northern region of the peninsula is now considered a subpolar climate that no longer has year-round sea ice, and it's a warmer and wetter climate than it used to be. Versus the far south region is still considered a truly cold, dry, polar climate. And then we have the south that's kind of our transition zone. And then we're also interested in understanding this coast to shelf to slope gradient, where the shelf is considered our most productive region. It's where all the whales are foraging and whatnot, and the penguins live, versus our slope region, which is more offshore and more influenced by inputs from the Antarctic circumpolar current, for example. We also have a moored sediment trap that's located off the shelf, and I mention that because that'll be important for my second question that I'll answer today. In terms of macro zooplankton collection for collecting things like pteropods, we use a big 2-meter framed net shown here and deploy it off the back of the ship. And then we collect all of the zooplankton in what's called a codon shown here. And when we're trying to collect animals specifically for experiments, like I'll talk about in my third question, sometimes we will do important dances to elicit the pteropod gods to bring us more pteropods. And I wouldn't say it always works, but it does make for a more fun time on the back deck. So moving on to some of the results, I utilized 20 years of pteropod data to develop these anomaly plots. And so any bar below the 0 line here indicates below average abundance for that year, versus any bar above the 0 line indicates above average abundance for that year. And what we see is that since 1993, there's been an oscillating pattern in pteropod abundance over time, but no long-term directional change. And we found that climate oscillations were actually an important control on pteropod abundance. This figure here shows the MEI, or the multivariate IMSO index, on the x-axis, and the Limacina pteropod abundance anomaly on the y-axis. And we see that a more positive MEI, or an El Nino year, corresponds to lower pteropod abundance. Conversely, a La Nina year corresponds to higher pteropod abundance. And typically along the peninsula, La Nina affects the wind conditions along the peninsula such that lower sea ice typically occurs that year. And so overall, we're seeing that pteropods are typically more abundant in lower sea ice La Nina conditions. We also saw that sea surface temperature was an important predictor, such that higher sea surface temperature corresponded to higher Limacina abundance anomalies the year after. And so overall, we see that pteropod abundances were best supported by ice-free warmer waters, which is actually sort of good news for these animals, in that despite this long-term warming we've been seeing along the peninsula, these animals are actually hanging in there and doing okay for now. Moving on to my second research question, have Limacina phenology patterns shifted over time, and what environmental conditions control these patterns? And so to answer this question, I utilized a sediment trap, which I mentioned is located off the shelf of the LTER sampling region. And for scale, I'm standing here, pretend I'm not in my slippers. And this is the sediment trap, and it's a giant cone that basically is catching any detritus, marine snow, zooplankton that happen to fall into the trap. And we deploy it at about 170 meters below the surface, and has a series of bottles that that sample and rotate around like a carousel, and they rotate about every 2 weeks to a month, depending on how much flux we think we're going to get. And I analyzed cups from the sediment trap from 2004 to 2018. And what's great about this sediment trap is that it collects data year round, as opposed to our research cruises that only go out for a month in January. And what's also great is that pteropods happen to fall into the sediment trap quite often, because one of their defense mechanisms is to retract into their shells. And we think what happens is that pteropods hit the sediment trap and they're like, oh no, and so they retract into their shells, fall into the trap. And it creates this really wonderful time series for me to analyze. So that's what I'll be showing you today. And in terms of some of the methods we were using, we were picking out pteropod shells. These are what some of them look like from the sediment trap, and then measuring their lengths. And I had a ton of work from an undergraduate from William & Mary, Colleen McBride, helped me with this. And I figured out she measured about 10,000 pteropod shells over the course of 2 years to help me with this project. So thank you, Colleen. In terms of some of the results, I'm going to show you some size frequency histograms. So we have length here on the x-axis, and then the count of those lengths on the y-axis. And this is just for 1 year, 2017 to 2018. But what we typically see is that most of the pteropod shells begin to show up in May or June. They are fairly small, less than 1 millimeter in size, and they grow throughout the winter season. And this was really exciting to see, because we actually don't have any documented evidence that pteropods grew throughout the winter when there's not much food. And then what we see is that there's not a lot of pteropods in the sediment trap in the summertime, which is in January in the Southern Ocean. And this is because we think they're feeding in the surface waters. We do occasionally get some larger adults that we think may have just died off due to predation or whatnot, but most of the time we don't collect any pteropods in the summer. And then what we think happens is that they spawn, these adults are spawning, and then autogenetically migrating into deeper waters, which is something that commonly happens for polar zooplankton. And then those offspring are showing up in the sediment trap again around May or June each year. And so one of the things I did was look at whether this time of appearance in the sediment trap was changing over time. And so this figure here shows the day of year that the pteropods began to show up in the sediment trap over time. And we see that there's a lot of variability with no long-term directional change. And so overall, we see that the pteropod phenology in terms of their appearance in the trap has remained stable. We see that this variability corresponds to sea ice retreat. So this figure here shows sea ice retreat on the x-axis as an anomaly, and the time of appearance on the y-axis. And we see that typically earlier retreat corresponds to earlier appearance. And so while there's been no long-term directional change in the phenology of pteropods, we see that it is tightly corresponded to sea ice. And finally, looking at changes in environmental conditions related to pteropod metabolism, one of the effects of this long-term warming that we've seen along the peninsula is changes in chlorophyll A or phytoplankton biomass. And so this figure here shows satellite chlorophyll over 20 years. And by comparing that time period, they've seen that in the northern part of the peninsula, there's been a decline in chlorophyll A shown in blue, versus in the southern part of the peninsula, there's been an increase in chlorophyll A shown in red. And what we believe is happening here is that in the northern part of the peninsula, as I've mentioned, there's no longer year-round sea ice. It's a subpolar climate. The temperatures are increasing, and in concert with decreased sea ice, we're seeing increased mixing in this region as winds blow over the seawater more and more. And this mixes the phytoplankton below their critical depth, such as they can't photosynthesize and bloom. And so overall, we've seen a decline in their biomass over time. Versus in the southern part of the peninsula, where year-round sea ice is still present, increases in temperature and associated in some declines in sea ice have led to occasional releases from light limitation for these phytoplankton. And so essentially what we see is that periods of sea ice melt with increased light limit, decreased light limitation lead to increases in chlorophyll A biomass over time. And so I wanted to see how these differences in temperature and food might be affecting pteropod metabolism. And so to do this, I set up a multifactorial experiment where I did many pteropod dances to collect pteropods in my net, and then also did CTD casts to collect what I call high or ambient chlorophyll A, and then I would dilute some of that water down to get a low food treatment. I then would acclimate the pteropods in seawater at a high temperature treatment of 4 degrees C, and then a low temperature treatment or an ambient temperature at 1 degree C, and as well as their corresponding food treatments for 2 hours. And then I would run 12-hour incubations and measure their metabolism. And the way that I was measuring their metabolism was through their respiration and excretion. And so their excretion was measured by their oxygen uptake rates. And I did this using a fiber optic cable that basically shines a red light, like this little laser pointer here I'm using. And it's able to measure the concentration of oxygen molecules in this bottle where I have about 5 pteropods in. And I would do that every hour for 12 hours. And then at the end of the experiment, I would measure their excretion by filtering out all of the chlorophyll and the pheopigments from the bottle, and then freezing the filtrate and measuring back at VIMS different dissolved nutrients such as dissolved organic nitrogen and carbon, ammonium, and phosphate. And just to briefly go over some of the results, I found that temperature was an important factor in increasing pteropod metabolism, but not food. And so this figure here shows temperature on the x-axis and then respiration rate on the y-axis, and the corresponding food condition here in the color bar. And what we found was that increased temperature typically corresponded to increased respiration rate, but food did not have a significant effect. And this was also found for most of the excretion variables that I measured as well. And so overall, we see that along the peninsula, temperature may be a driving factor of pteropod physiology on the short term, but not as much food conditions. And so one of the main conclusions I make of my dissertation is that we could consider pteropods not only bioindicators of ocean acidification, but of climate change in general along the peninsula, because we see that they're responsive, in addition to ocean acidification, to changes in sea ice, particularly related to their phenology, as well as temperature related to their long-term trends and their physiology. And we see that on top of this, there's subdecadal oscillations such as La Nina and El Nino that are affecting pteropod abundances over time. And so I argue that we can really use them to measure multiple factors along the peninsula, not just ocean acidification. And this is why I think pteropods are so terrific. Shifting gears now and moving up to the northern hemisphere to Narragansett Bay, we're in a temperate estuary that has been exhibiting long-term increases in temperature, as well as declines in nutrients and increases in water clarity. And some of the effects on these changes has been an expansion of warm water species residence periods, particularly for warm water fish, such as the butterfish shown here. We've also seen that warmer winter and spring temperatures have corresponded to higher abundance and duration of jellyfish, such as Meniopsis ladii, shown here. And we've also seen a long-term decline in summer chlorophyll A. But this data set only analyzed data from 1972 to 2006. And we have over 60 years of chlorophyll and nutrient data available to us through the Narragansett Bay long-term plankton time series, which is what I've been working with. And to give you some more context for this time series, it's located in the lower west passage of Narragansett Bay, shown here. And to orient you, we have the Graduate School of Oceanography, where I work remotely, shown here near Rhode Island Sound. And since 1959, students and staff have gone out weekly to collect measurements for phytoplankton and zooplankton counts and biomass, nutrients such as ammonium, phosphate, nitrate, nitrite, and silicate, and water quality measurements such as temperature, salinity, and water clarity, as well as weather data. And some of these data are publicly available at this website shown here, and people have been analyzing those data, but no one's actually been able to convert all the data into usable metrics to analyze for the full time series due to differences in data collection and whatnot over that time span. And so one of the first steps of my postdoc was to spend time harmonizing 60 years of these data, and that meant I really was going into a closet and trying to find the archived printed data from 1959. But I was able to find a good amount of data to harmonize. And so this allowed me to ask questions like, what are the long-term trends in nutrients in chlorophyll A in Narragansett Bay? Has the timing and duration or the phenology in phytoplankton blooms changed over time? And what environmental factors are related to bloom phenology trends? And as some of you may imagine, harmonizing 60 years of data, I wasn't able to find or harmonize data for every week of every year of the time series. And so there's an issue of missing data. And one of the ways I dealt with this was to work with statisticians at URI to develop dynamic linear models. And basically what this allows me to do is to interpolate missing data with uncertainty. And it interpolates based on using existing structures of the data set in hand. So as an example, we have the raw or observed data for ammonium shown from the time series location from 2004 to 2019. And there are clearly some gaps in this data set. And so by using the dynamic linear models, we can then interpolate those missing data so we can get a consistent time series throughout this time period. And another great thing about this model is that it allows us to separate the long-term trends shown here versus the seasonal components shown here so that we can analyze them separately. And this is particularly important when we start to try to understand long-term change in relation to climate. And we also are able to look at any parameter and component and how it varies with time. And this is particularly important when we're trying to answer questions about phenology, where the timing might actually be shifting. And for the purpose of today, I'll be showing you a mix of both the observed and the model data. And I'll be letting you know when I'm working with both, or with one or the other. And so to look at the first question, what are the long-term trends in nutrients in chlorophyll A? And I first answered this question by looking at average annual concentrations of nutrients throughout the time series. And I note that these data are observed values from the time series and not the model data, so that you can get a better sense of the variability in nutrient concentrations over time, which are shown as 1 standard deviation above and below the mean. And we see that phosphate concentrations have significantly declined from 1 to 2 micromolar to less than 1 micromolar since 1959. And we see a similar declining trend for ammonium, where there's been a decline from about 2.5 micromolar to about 2, although this is a rather weak decline, and it is significant. And we see that for nitrate, nitrite, there's been an oscillating trend in their concentration over time, with particularly stark differences in the 1980s versus the 1990s. And conversely, we see that there's been a long-term increase in silicate, such that in the 1950s, concentrations were around 10 versus around 15 today. And so overall, we see that there's agreement with previous findings that there's been some long-term declines in nutrients, as well as some increases in other aspects like silicate. But we don't see quite as a pronounced decline as some other studies have shown, mainly because our location is located near the mouth of the bay, where there's probably more marine inputs and less so from wastewater treatment plants located further up in the bay. Moving on to chlorophyll A, again, I'm using observed or raw data here. We have chlorophyll on the y-axis and year on the x-axis, and the time series for chlorophyll A began in 1968. And we see that there's been a long-term significant decline in chlorophyll A. And I was also interested in seeing if the intensity of these chlorophyll blooms over time has changed. And I looked at the annual max concentration of chlorophyll A to identify this. And again, we saw a long-term significant decline in chlorophyll A intensity. So overall, we see that the mean and the max of chlorophyll are declining over this extended time series. Once I had understood the long-term changes, I then wanted to look at more seasonal differences in the chlorophyll A distribution over time. And so to do this, I used the model dynamic linear model data, and these represent average annual patterns of chlorophyll A for each decade of the time series. So from 1968 to 79, all the way to 2010 to 2019, shown in this light green line here. And we see overall that there's been a bimodal pattern in bloom distribution or chlorophyll A over an annual cycle, but that this pattern has changed significantly over different decades, such that in the 1970s, there was a very pronounced winter, spring, and summer, fall bloom, whereas in other decades, like the 90s and 2000s, there was a very depressed to almost no winter bloom and a gradual summer bloom later in the year. We do interestingly see a slight uptick in chlorophyll A distribution in the last 10 years. And so this then allowed me to answer questions about whether the phenology or the bloom timing has been changing in relation to these shifts in chlorophyll A distribution. And so looking at this question of phenology, I needed to define metrics to quantify bloom start and bloom's end and bloom duration. And so to do this, I looked at a suite of different phenology metrics and found that this method by Siegel and colleagues best characterized the blooms in the Bay. And they use what's called a 5% median threshold. So for each year, as an example, this is 2016 chlorophyll A concentrations. I would take the median chlorophyll A concentration for that year, and then the first time that the chlorophyll A is greater than 5% above that median, which is shown here by the purple line, that would be identified as the start of the bloom. So for 2016, that's around week 5. And then when that chlorophyll value crosses below that 5% median threshold, that would then be the end of the bloom. And the duration would then be the difference between the timing of the bloom end versus the bloom start. And I broke up these phenology metrics by season, such that we have winter phenology metrics from about January to the end of March, and then summer phenology metrics for weeks 20 to 35. So I determined when the blooms started in winter and when it reached its max and how long it lasted in winter. And I found a lot of variability in these different metrics over time. I note again that all these data are based on observed chlorophyll data as opposed to model data, so that we can get a better sense of the bloom variability over time. Overall, I found no long-term directional trend in winter bloom start, max, or duration. So I've summarized these data into box plots to get a better sense of when the blooms are starting their median start and max and duration over the time series. And so overall, we see that blooms are typically starting in Narragansett Bay around week 3, so in the later part of January, but they can start anywhere between the first week of January to the end of March. And the bloom maximum is occurring around week 5 or in February, but again, it can range anywhere from January to April. And the duration is lasting around 3 weeks. And then if we look at the summer bloom phenology, we see, again, no long-term directional change in the bloom start date or initiation or the bloom max. We see that the blooms are typically beginning around week 24 and they're reaching their maximum a week later in week 25. We did see a significant difference in the bloom duration, such that the blooms, the first 20 years of the time series from 1968 to 1994 were beginning around week 6, whereas in the past 20 years, they've been shorter, lasting around 3 weeks. And this is just to show you the actual data and the long-term modeled fit that's been occurring over time. But again, we do see that there's a lot of variability, which has been a wonderful characteristic I've come to understand working with estuaries. In terms of bloom frequency, I wanted to see if the number of blooms was changing over time, and we saw no long-term change in bloom frequency by season, but we do see that there are significantly more blooms in summer than there are in winter. So typically there's around 3 blooms in winter and around 4 to 5 blooms in summer, for a total of about 7 blooms per year, but overall no long-term change in the number of these blooms over the time series. And overall, this indicates that there's a lot of variability in the bloom phenology in Narragansett Bay. And this corroborates some of the work that was found by Ted Sameda, who originally founded this time series 60 years ago, in which he said the same thing in 1998. So in some ways, we're just seeing more and more variability in this region. And Scott Nixon also published some work noting this as well, although he was using phytoplankton counts as opposed to chlorophyll A biomass, so a slightly different metric. But overall, we can see that with 50 years of chlorophyll A data since the 1960s, we have a much more holistic picture now of the bloom dynamics as well as the nutrient concentrations and how they've changed over time. And so this moves to my third question of what environmental factors might then be related to this bloom phenology variability. And I used a dissimilarity-based method called BioM, which basically allows me to use multiple parameters at a time to look at how they might all be influencing bloom phenology. And so I used a suite of different metrics, including climate indices like the North Atlantic Oscillation and the Gulf Stream Index, mean winter and summer nutrient concentrations, mean winter temperature and salinity, as well as water clarity and light. And by doing this, I then was able to use BioM to identify the best model fit to explain winter bloom start, max, and duration. And one of the factors that was prevalent in all of these models was temperature. So as an example, we found that higher mean winter temperature corresponded to shorter blooms. And in the summertime, we found that multiple environmental factors affected the summer bloom phenology, including climate indices, salinity, and nutrients. And so as an example, we saw that higher mean salinity typically corresponded to a later bloom start date in summer. And so overall, we see that multiple environmental factors and not just one driving factor, such as long-term temperature changes, are influencing this bloom phenology in Narragansett Bay. And so to conclude, we see we now have 60 years of continuous data of chlorophyll A and nutrients for Narragansett Bay. We see that the blooms are declining in magnitude and intensity, but that their timing is staying stable over the course of this time. And I wanted to showcase this in a more creative way. And so I did this by taking the data that I've shown you and creating these giant plankton murals. And I had an opportunity this summer to collaborate with the Rhode Island School of Design in which I utilized the actual data that I had shown you earlier, looking at these decadal changes in chlorophyll A distribution, and used them to make these patterns that you see over the different panels in the different decades. And then I took phytoplankton microscopy images from the time series and learned how to convert them into illustrative graphics so that I could then create repetitive patterns of on these panels to show how communities and phenology influence the phytoplankton in this time series as well. And so this is a really cool way for me to think about my data in a different way that I hadn't done so before, and also use it as a science communication tool. And so building off of my earlier conclusions, one of the things I think might be happening, or that I'm thinking about, is whether this variability that we see in phenology might actually aid in the stability of the system, and whether this large variability we see in the timing of the blooms is actually helping it to cope more with these long-term changes in temperature that we're seeing. And that's something I'd like to look more into with my research. We also saw that phenology correlated with multiple environmental factors, such as temperature relating to shorter winter blooms and higher salinity corresponding to later bloom start dates. And by understanding phytoplankton bloom dynamics, this aids in addressing ecosystem-wide changes with ocean warming. So by understanding what's going on with the phytoplankton, we can then better connect what's going on with the zooplankton as well as the fish in the region. And one future direction I'd like to take with this research is looking more at species-specific changes and whether shifts in timing and abundance of phytoplankton community composition might help to explain these decadal trends we see in chlorophyll A distribution over time. So I'm currently working with Tatiana Rainierson at the Graduate School of Oceanography to utilize the species counts that have also been collected as part of this time series to see whether different abundances of phytoplankton, such as Thalassiocyra shown here, might correspond to these changes that we see in bulk chlorophyll over the time series. So with that, I would like to thank my funding sources during my PhD, which includes the NSF Office of Polar Programs and the Virginia Institute of Marine Science. I was working with Deb Steinberg in her zooplankton ecology lab at VIMS, which was a really great experience. And I'm currently funded by the Rhode Island Coastal Ecology Assessment Innovation and Modeling Grant, which is funded by RI EPSCoR. And I'm working with Tatiana Rainierson in her phytoplankton ecology lab, and I began my postdoc in January 2020, so have spent most of it in my house and on Zoom. So it's been a bit of a different start to my postdoc, but I am really enjoying it here, and that's one of the reasons I've been applying to some grants to try to stay a little longer if I can. But I'd also like to thank the Graduate School of Oceanography, particularly Ted Sameda, who started this time series, as well as David Borkman, who worked with the time series for a number of years and has helped me quite a bit in getting a sense of where things are located. And then Jacob Strock and Gabino Pugioni, who are the statisticians I've been working with at URI to do the dynamic linear models, and all of the graduate plankton assistants that have gone out every week to continue collecting and building on this plankton time series. And with that, I would like to take any questions. Thank you very much.
All right, thank you so much for that presentation. A lot of information there, and hopefully we have questions from the audience. You can raise your hand or type in the chat, or just go ahead and unmute, and I'll help field the questions. I had one question just to start out with. On the Narragansett time series, the nutrient trends, I was wondering if there's changes in the estuarine circulation that's happening, as well as something happening in the ocean. So did you look at like a salinity normalized trend, or was that just the total observed nutrient?
Yeah, that was the total observed. I haven't thought about doing the salinity normalized. That would be something to look at. I know, at least in terms of the circulation of the bay, it's thought, and this is more regarding plankton communities, that it typically water enters in from Rhode Island Sound into the East Passage, which is the other side of the bay, then circulates around from Providence and comes back down to our time series site. So it does make for sort of an interesting interpretation of what our snapshot represents over time. But I haven't thought much about the circulation in that way. So that could definitely be something to look into more. Yeah, thank you.
Okay. Xiaodong, did you have a question?
Yes, I do. Hi, Tricia. On your Antarctic study, you found that sea ice had influenced that terracotta distribution. Exactly how sea ice, in your mind, affect that? Is it through the
So,
temperature or nutrient or the food?
so I think what's happening there is that the timing of when the sea ice is retreating, because the location of where the sediment trap is seasonally covered by sea ice. So I think that when the sea ice retreats earlier, it's allowing the pteropods to then feed more and grow more. And so then they're appearing in the sediment trap earlier because they're spawning sooner, partly because maybe they can have more access to food.
But you also found the food is not a factor, right?
Yes, and I think I didn't have a ton of time to go into this, but I recognize I'm working on very different time scales. So the metabolism experiments I did were, 12 hours versus the phenology work I'm doing is on the scale of years. So thinking about how that might, food might affect those different time scales is something to consider. But you're right. I mean, I think it could be other factors as well, not just food. And unfortunately, we don't have any environmental data that's collected directly on the sediment trap to really get a better sense of like what the circulation patterns are, anything like that. So I think that could be a cool thing to try to do for a future grant.
Okay. So in your sediment trap, you collect the theropod. When you collect them, are they still alive or they died? That's why they settle down there.
Yeah, so I believe that a lot of the ones that we collect are alive when they hit the sediment trap because there's still a lot of tissue in the animals. So the image I showed, the yellow stuff was its tissue inside of the shell, versus occasionally we would collect shells with no tissue in them. And I think those are actually, part of the particulate inorganic carbon flux of shells that are pteropods that have been eaten, and then the shells are sinking because some of their predators actually will suck the tissue out of the pteropod shell and the shell will sink. Okay. So that's one of the reasons I think that we're working mostly with live pteropods. Once they enter the sediment trap, we have a preservative in them that fixes them and kills them. But I think that's one of the reasons that it's evidence for ontogenetic migration, because we think that the pteropods are ontogenetically migrating down to the 170 meters. And so that's why they're showing up in the sediment trap still being alive, so to speak.
Great, thank you.
Yeah.
Stephen Howden.
Hi, Tricia. I enjoyed your seminar. I don't know if I missed it for these Narragansett sampling. Was it, is that off the dock?
That is technically slightly north of the Graduate School of Oceanography. So they do take a small boat to the location, and it's about an 8-meter depth sampling location. One
thing I was wondering is about, I don't really know, is how heterogeneous phytoplankton are in the bay. And so if you're going out and sampling one week and the wind's blowing one direction versus another, might result in some of that more high frequency changes that you see that might affect your bloom timing, if it happens to go below the 5%. And I was wondering if you could use from the ocean color period kind of get an idea of how homogeneous or not the phytoplankton blooms are in that area, and how representative, given weekly sample would be.
Yeah, I haven't actually thought about looking at the remote imagery for that. Someone at URI, Colleen Mao, she does quite a bit of work with remote sensing, as well as she has an in situ flow cytobot that allows us to collect continuous measurements of phytoplankton. And so one of the goals I have is to maybe compare our weekly counts with that of the IFCB, which is literally sampling every 15 seconds, which might give us a better sense of the differences, or how well a weekly sampling frequency really captures the change in the community dynamics. The region there is considered to be fairly well mixed, but I recognize, and this is one of the limitations, that we decided to pick time over space as our sampling scheme for this project. So we are missing potentially some of differences in distribution compared to other regions of the Bay. But there have been other studies done by the Department of Environmental Management and whatnot, more specifically on harmful algal bloom species, that has shown that they are relatively evenly distributed in the Bay. And I know, at least when Ted Sameda first began the time series, he felt that weekly observations would be reasonable in terms of catching that bloom variability. But you're right. I mean, really, if we wanted to try to pinpoint down that accuracy, we would need something at a higher frequency to really capture that. But checking the remote color sensing would definitely be a good way to verify. So thank you for that suggestion.
I actually had a follow-up question. So you found there wasn't a big change in the timing of things in your time series. Is that actually consistent with what you showed at the beginning with the New York Times seasonal changes happening more in the southern US? Does that make sense that on land would be similar to near the coast?
Yeah, I haven't thought about that before. I know, at least, especially in estuaries, there hasn't been a ton of work looking at phenology in blooms. There was some work done in the Chesapeake Bay a couple of years ago, and they found some of the blooms were occurring earlier, but there was sort of this complex relationship with the concentration of ammonium and like the relative, I guess, hypoxic conditions in the Bay, and had to look back at that paper. But I think it is something that the field of marine phenology, I feel like, is a lot younger compared to that of terrestrial. So looking more into that, I think, could be or provide a useful perspective.
Okay, any other questions?
No, I have a question.
Go ahead.
In your linear dynamic model, I see that after you do that interpolation, and the interpreted value, even for those days or years that you did have data, they're kind of different, right? It's kind of this interpolation, you replace all the data regardless if you have or not.
Yes, that's true. Which is one of the reasons that I ended up showing some of the raw or observed data, because I was still and am still interested in trying just to understand what the actual magnitudes of change are over time, which you do lose a little bit when you do the modeled data. But I think in terms of trying to understand some of the broader long-term trends, it's more statistically robust to look at it in this modeled way. And I didn't show those figures as much today, but it does often corroborate what I'm finding with the raw or observed data most of the time.
Okay.
All right, if that's it for now, let's all send our good vibes up to Rhode Island and thank Patricia again for the presentation. And there's a few people that wanted to stay on to talk with her a little bit more, and anybody else is welcome to stay in the room for a little while.
Awesome. Thank you so much.
Thank you.